Now, some of the major reasons for Mendel's success as to why Mendel was successful in putting across the laws of inheritance which are valid even today. So, some of the major reasons are he studied inheritance of one character at a time. So, he made his work very systematic. He considered one character at a time. For example, in monohybrid cross he considered the character of height. In dihybrid cross he considered the character of the shape of the seed and the color of the seed. So that this is how systematically he studied the characters, and that was one of the contributing factors for his success. And he selected only 7 pairs of contrasting traits or 7 characters in the pea plant. I think the ones which were very easily identifiable were him. Obviously made his study, his observation and tabulation of results much more simpler and easier to understand. Another very important thing is he applied a lot of statistical and mathematical logic into his work, although this was not widely accepted by the biologists of that time. But of course, using statistics and using a very large sampling size which gave credibility to his data was another reason why Mendel was hugely successful in his work. And luckily, whatever characters Mendel chose, they were all located on different chromosomes. Especially, I would like to mention about the dihybrid cross. The genes controlling the shape of the seed and the color of the seed were not located on the same homologous pair. As you can see, the 7 characters which he chose were restricted to only the 4 chromosomes out of the 7 pairs of chromosomes in pea plant. In pea plant, all of you please remember, the diploid number is 2n is equal to 14. That means they have 7 pairs of homologous chromosomes. Out of the 7 pairs, the first pair, the fourth pair, the fifth pair, and the seventh pair had the genes for all the 7 characters that he considered. And he was lucky because the characters that he worked with, especially in the dihybrid cross, they were not linked, as in they were not located on the same homologous pair. For example, he got an F2 generation ratio in the dihybrid cross of 9 is to 3 is to 3 is to 1, and he saw that the character of the shape is not always tied up with the character of the color. If you remember, you're not always getting round and yellow seeds, you're also getting round and green seeds, isn't it? Similarly, you're not always getting wrinkled and green seeds, you're getting wrinkled and yellow seeds as well, isn't it? So basically, he identified that the character of seed shape is not always tied up with the character of seed color. And that was because, luckily, when he was conducting his dihybrid cross, the genes controlling the shape of the seed and the genes controlling the color of the seed were not located on the same homologous pair. And when we say that they were not located on the same homologous pair, that means they were not linked. And that's why he got a classical dihybrid ratio of 9 is to 3 is to 3 is to 1. So these are some of the reasons why Mendel's work was hugely successful, and he is rightly referred to as the father of genetics.